Thank you very much. So I guess I'm the only alien here to the take research infrastructures, but we thought it's good that we start talking across the corridor and learn more about what kind of services are being provided in other facilities of Masaryk University. So like the Recetox Research Center runs also the Recetox Research Infrastructure, which is at the same time coordinating hub of the IRIN infrastructure, which is the European S3 infrastructure, the only one that is coordinated from the Czech Republic. This is the description of the research infrastructure that operates in the bridge between the environment and health. And as you can see, the things that we are doing are relevant both for the environmental research and then to the biology, health, and food research as well. I like to use this picture because it shows nicely where we are. We are trying to connect the environmental problems with the societal and health problems. So basically looking at into the environmental factors, how the society affects the environment, how environment affects the human health, how the societal issues are also affecting the human health. And all of this is hidden in the word human exposome, which means all the factors that are collaborating with the human genome and setting up the human health and well-being. I said that the IRIN research infrastructure is in the European roadmap since 2022, but Recetox research infrastructure is a part of the Czech national roadmap already since 2011. It is a single-sided infrastructure, so we don't have partners there, and it has several facilities. The oldest one is the facility of the accredited laboratories. Petra Přibylová, who is the head of our laboratories, is right there. Then the second facility are our monitoring networks, because we are running a lot of air and water monitoring networks. If you look at the map here, all the blue dots are the monitoring sites that are being operated by our center. Then we also do the population studies. I will talk more about our population studies and human cohorts later. And then, of course, the information platform. As you said, you can't do anything without the data. I dare to say that we were like 15 years before the EOS and we started investing into development in-house and later national and international infrastructures already some 15 years ago. So I said that we speak about human exposome, which is all the factors behind the human health. There are basically two approaches. You either look into all the chemicals that are in the food and air and water and cosmetics, and then looking how they affect the human health. This is the so-called external exposome. Or we start from the top and measure the chemical directly in the blood and see how the exposure to various environmental factors is translated into the biological parameters. But since both the biomarkers of exposure and biomarkers of effect are mostly using the same technology because it is mass spec based, so we can link them together and talk usually about chemical and biological profiling. So then this is the picture showing that the genotype is somehow reflected or deciding on the phenotype, and the science of it is called functional genomics. In the last 5 years, we speak a lot about functional exposomics, which is the natural complement. So the genotype together with exposotype decides on what is our final phenotype, but we also have to think about the lifetime. So like the genotype is not something static, but it develops throughout the life course. The same approach we have to take to the functional exposomics. What we are doing is we are investing a lot of the new things into development of the tools for functional exposomics. So as you said, our original services were based on measurement of anthropogenic chemicals, the organic chemicals and toxic metals and how they accumulate in the natural matrices in air, water, soil, food, name it. But then we also started measuring the chemical components in the human tissues, and then it was only a step from looking into targeted and non-target metabolomics and proteomics. As you know, this is a big investment, but especially if we move towards the non-target metabolomics, and there is a still big depth in the whole field of metabolomics because things are not well documented. There is not very strong QA/QC behind it. So we are investing a lot into building open access resources for metabolomics and exposemics, including public SOPs and spectral libraries, public data processing pipelines. In that, we collaborate with several other infrastructures, including Elixir. We are using Elixir Galaxy pipelines to build the new pipelines for assessment of data for both targeted non-target GC and LC metabolomics. So if we speak about the services of the accredited laboratories, you can see the whole list of the chemicals that we are analyzing. Pretty much anything from organic and inorganic. It's not only about fluorinated, brominated, or perfluorinated compounds, but also plasticizers, the UV filters, masks, mycotoxins, cyanotoxins, micronutrients, and all of this. But as I said, we are also now investing a lot into the targeted analysis of the metabolites. The goal is to provide also the services for the clinical studies, not only population studies. So we are clustering into the different clusters of the biomarkers that are being used for different clinical studies. When we talk about the analysis of endogenous chemicals, there are just few examples of the targeted metabolomic groups. Some of them were developed for our collaboration with the people looking into the human exposome, so identifying the metabolites of the microbiome in the human blood. Some were developed more for immunity studies and so on. But the biggest investments are in this era, which means really looking into chemical and biological profiling via high-res resolution. So we are using both GC and LC Orbitrap platforms. We have different setups, partially automated, and we are interested in the new collaboration. I already mentioned that besides metabolomic platform, we also look into the human microbiome. So we are also offering a lot of services in the analysis of in the area of analysis of the microbiome, including preparation of the libraries for short-term metagenomic sequencing and detection of DNA. We are working with different type of microbiome samples. It could be fecal, skin, nasal, dermal, all kinds of the microbiome that can be analyzed in the laboratories. So very much about laboratories. I already mentioned that we apply our analytical methods for different types of either environmental or human studies. I said that we are operating the oldest history is the operation of our air monitoring networks. We work as the collaborating center of the United Nations Environment Program that is responsible for the international conventions protecting people from the chemical compounds, from the chemical risks. So that's why we set up our networks in Europe, Africa. We collaborated with South America, with Asia. Those are just the illustrations of some of the results about the trends of the chemical compounds in the ambient air in the last 10 years. We are doing the same activities with water, and we are not only building the monitoring networks, but also information systems. So our systems can be used not only for presentation of our internal data, but also external data. And this GMP web page or information tool is a major tool of the UNEP that is being used for the global monitoring of the toxic chemicals. So it means every 6 years there is a global data collection. We are using our own systems for the people to upload their data. They are harmonized and brought together and then presented. There are different ways how you can present the availability of data. You can use the maps or the graphs showing where the data are coming from, what is the time scale. We can do the summary statistics, or we can even do the trend analysis. And this type of of the software can be developed, for specific studies on demand. You can use it to present your own study to make it more visible, to make them accessible for some, global comparisons. And the last thing is our population cohorts. When I speak, then you have to understand the difference between the cohort and cross-cutting study. Cross-cutting study are typical clinical studies when you at one time point you collect the sample of certain population. While if we are talking about longitudinal cohorts, it means that you follow specific populations throughout the lifetime. So our oldest cohort is the LSPAC cohort that was recruited in 1990, and it was part of the pan-European study that was originally organized by the World Health Organization, the European branch in Bonn, that wanted to recruit 40,000 pregnant women in Europe and follow the children until the adulthood. So at the beginning, some 12, 15 countries got involved in the pilot, then a little less initiated the study, but there were only 2 countries who really managed to follow the kids until the adulthood, which was the coordinating center at the Bristol University and then the Masaryk University. So we have one of 2 cohorts that made it to the end. In the Czech Republic, it was all the kids that were born in Brno between 91 and 92. It was altogether about 6,500 children, plus another 1,500 in Znojmo for comparison of bigger and smaller city. And those kids were really followed until the adulthood. The problem was that they had a lot of data, pediatric data, socioeconomic and environmental data, but very limited biological samples, only the DNA samples from saliva collected during the puberty of the children. When we took responsibility for the cohorts 10 years ago, we first had to digitalize all the available data, which was about 300,000 questionnaires about diabetic that we need to bring to the electronic form. Based on it, we prepared the protocols for the follow-up studies. So today we are reassessing all those kids. They are now young adults, they are 30 plus. And we are reassessing them, collecting the biological samples from them, but also in parallel, we are also recruiting the pregnant women for the second generation of the birth cohort. We currently have about 2,500 of mother-child pairs that have been already recruited. Again, in the biobank, we have the samples from the pregnant woman and then from the children, the cord blood, urine, meconium, the buccal smears, I think a few others, including dry blood spots. And then in collaboration with University College London, we are also responsible for the HAPPY study, which is the Central and Eastern European Aging study that was recruited in 2000 in the Czech Republic, Poland, Lithuania, and Russia. Out of it, some 7,000 aging people were recruited in the Czech Republic, and again, they've been followed for the last 20 years. Currently, we again are reassessing those people after 20 years. And there are a few other arrows showing that this region is really rich with the population studies. The first mother-child cohorts were recruited already back in the 60s. And then there is also the cardiovascular cohort that was recruited by ICRC, which is the reproductive age cohort. And then, of course, if we wanted to work with the biological samples, we had to invest into the construction of the biobank. The Recetox biobank is one of our core facilities. So again, it can be used for providing services. We can offer the storage place for the samples, but especially all our sample collections are open access resource. So it means people who are interested in the research or collaboration using the samples and data from the population cohorts can ask either for an access to the data or access to the samples or some types of the collaborative study. From CELSPAC, we have a long-term collaboration, for example, with the neuroscientist Klára Marečková. She's running her neuro research using the CELSPAC cohort of young adults. So these are just few pictures from the biobank, it is -1 and -2 floors, so it is below the ground, and this is how the automated technologies look like. For those who are working with the biological samples, you know that the quality of storage of the samples is absolutely crucial if you want to analyze something and publish something reasonable. So this is why we invested not only into the automatic systems for the biobanking with the control quality, but also automated systems for the robotic analysis of the samples. So, of course, what we built is not only one population cohort, but the whole platform for the population studies. So it means it can be used for various studies, and those are just examples of other large studies that we provided our infrastructure for. The most important is Recetox study, because during the COVID pandemic, Czech Republic didn't really have any institution that would be able to run the national seroprevalence study, because the Czech Health Institute doesn't really have the capacity and the network of the hygiene, so it's not functional anymore. So during that time, in collaboration with one of our insurance companies and the network of biochemical laboratories, we were able to recruit the cohort of 30,000 people, a representative sample across different ages and geographically covering the whole country. And those people were sampled 3 times before, during, and after the vaccination. So we have very nice cohort really stored for the future to see really the long-term impacts of the COVID pandemic and development of the neurological or metabolic and other disorders in the future. So these are just the services that we can offer through the CellSpec platform, which really includes collaboration in the study designs, protocols, the sample collection. We are currently collaborating, among others, with the SHARE infrastructure from the Czech roadmap that also has the large European aging study, but it is socioeconomic study, so it doesn't have the biological samples. They are very much interested in collecting also the biological samples from the existing cohort that has been followed for 15 years already. So we are now preparing the project with SHARE, where we will be taking care of all the biological part of the research. And of course, for those of you who are interested in working with the data or samples from the population cohorts, all our services, all data is available through our web page. And just a few words about toxicology. If we are assessing the environmental impact on health, we need to have the information about the chemical exposure, but then also about the toxicology. So we also have the small facilities that are housed in our research program, and one of them is mechanistic toxicology. And the key word for this work is adverse outcome pathway. I don't know whether you are familiar with the term, but it is basically saying that in order to understand better the toxicological effects of various chemicals, we really need to find the molecule-initiated event. And we need to follow the potential adverse outcome pathways. We have some 150 biochemical pathways in the organism, and we need to identify what is the initiating event that starts the adverse outcome pathway, and then map, the key events going to the adverse health effects. So it is pretty much the toxicological modeling, and we are trying to bring together the information about the different effects of different chemicals. And then, of course, we offer all the services of the toxicological laboratories, going much more from the testing on the animals to in vitro models, different stem models, the chips, and specific studies on different hormones, DNA damage, and similar. I'm done. Perfect time. Thank you very much.
Super interesting. So I hope the audience to have some questions for Jana.
With the boom of the AI technologies in the imaging, as people use it, do you also see it in your area, and which particular field is most most influenced by the advent of the AI?
Multiomics, because everyone speaks in the multiomics, but how many real multiomics studies have you seen? It's typically just linking genomics to proteomics because it's straightforward. Metabolomics is open question, and all those, exposures working with the non-target data, I can probably confirm, the protein is challenge enough. Metabolomics is multiple challenge, and looking into the chemical exposures means that you are looking into the concentrations 3 orders of magnitude lower than for endogenous chemicals. So it means it's a big pressure on the technology itself, big pressure on the data analysis. This is why we are investing a lot into those data processing pipelines. But still, that the trend of today is what they call the IT freezing. So it means rather than keeping the samples in the biobank, is running the non-target screening on different platforms and keeping the data, which of course requires huge storage capacity, but also huge computing capacity, including the development of new IT tools and bringing together, the data from non-target screening, from toxicological analysis, because if you can picture the graph of adverse outcomes pathways, you have more than 100 different pathways, multiple key initiating events, thousands or maybe hundreds of thousands chemicals, and each of them has more than one effect. So it's pretty complex metrics that you need to work with. And then, of course, also the data from the microscopes and tissues and imaging that comes together.
I may ask, what are the modes of access to your infrastructure? Is it based on some price model, on collaboration, mixed, project-based?
Anything.
And how
We
can
have,
you find out more?
you can find, the access point. Petra Rušičková, where is she? Over there. She's a manager of our open access. So the easiest way is just to drop her an email and talk to her, because we allow, the people to send the samples for analysis or come and work with us. We have a lot of international exchanges. I must say, we are real open access infrastructure. So the big portion of our capacities goes to the external users. We don't have too many users from Masaryk University. We do have some from our center, but many are really external. And especially now with the boom of the human exposome, Petra can say we have thousands of the samples where we are analyzing the biological samples from different European studies. So they either want to train their PhDs, so we would accept a PhD student for the semester to work with us, or we can provide the training, or they can send the samples. We also have our IT group, biostatisticians and bioinformaticians, because this is one of the two study majors that we offer. One of them is environmental health science, and the other one is in biostatistics and bioinformatics. So we do it, but it is mostly collected to our samples. We have one team who is developing the information system, one team working with metabolomics pipelines, the other one for microbiome pipelines. For the data from the cohorts, again, data are fully accessible, so you just need to reach out. If you go to the CELSPAC-CZ web page, you would find all the questionnaires, all tables with the existing data. You can just mark what you want and talk to the head of the facility how you need to harmonize data, and these data are coming into many European projects apparently. The same for the environmental samples. Environmental samples are fully open. You can't make the personal data fully open, but the environmental samples are fine. So you can even upload all the data to the page.
Please, can I ask, you mentioned many types of data and well structured. How large are in general, for example, the data sets about the cohorts or about the occurrence of different compounds in the atmosphere? Like about how many terabytes are you speaking?
don't ask me about terabytes. It's big, but we are using, as you said, different types of the data. So some like targeted data from the laboratory are small. They are basically large Excel tables, so that's easy. Even the targeted data are not very big, and we have no problem with storage. But what is the problem is really to store the high resolution data from on-target screening, and this is where we collaborate with Cerit a lot. You mentioned, how to collaborate with them. We do exactly the same. We are buying hardware for Cerit and paying the operational costs because we can't really manage it here.